Hello, my name is Sara Lucero. I'm a senior at Columbia River High School, and my experiment is a comparison of the soil organic carbon found in affected forestry around the Mount Hood area. In addition, I researched the effects of human impact on the forest, or on the future of forest management. I might need this. One of the blessings of living in our region is being able to enjoy the outdoors and the natural beauty of the Pacific Northwest, mainly a great expanse of forestry and the majestic Mount Hood. However, human interaction has led to major impacts in our area, specifically areas of wildfire and clear-cut deforestation. With this into consideration, I hypothesize that old-growth soil will have the highest percentage organic carbon, clear-cut soil would have the second highest percentage organic carbon, and burn soil would have the lowest percentage organic carbon. But this really does beg the question, what's so great about organic carbon? Well, besides being the most abundant element in soil organic matter, it is 60% the composition of the soil organic matter, and it plays an important role biologically, chemically, and physically. As far as chemically, it plays a part in the cation exchange capacity, which is the measure of soils, the ability to be a buffer using weak acids through processes such as nitrification. Biologically, it increases the retention of necessary cations used as nutrients, such as calcium, magnesium, and potassium. And physically, it adds to water retention during the drier seasons. Loss of organic carbon through fire and clear cutting degrades soil, and although soil does naturally recover over time, it could take up to 50 years for it to return to its natural state. The basis of my experiment is that carbon loss in affected soil is what degrades soil fertility. Here in detail is what happens when carbon dioxide is released when forests are affected. So old growth trees are what go through photosynthesis and uptake carbon dioxide through the air. Because of this and litter fall, soil organic matter increases, water retention increases, and generally carbon dioxide in the atmosphere decreases. When we have a loss of trees is where problems start to arise. Because the trees aren't there to undergo photosynthesis, and because of the combustion of trees themselves, there is a drastic increase of carbon dioxide in the air, as well as a decrease in soil organic matter and a decrease in water retention. The same effect happens for clear-cut soil where there are no trees for photosynthesis, so water retention decreases, soil organic matter decreases, and more importantly, carbon dioxide increases in the atmosphere. So how else does human interaction to climate change affect other local issues? Well, for the past 15 years, there has been a steady and noticeable decrease in snowpack on the mountain. The green line pointed out here shows the amount of snowfall on Mount Hood for the winter season of 2014 to 2015, and it's evident that we are on a steady decrease to another historic minimum in the past just 15 years. Less snow on the mountain means less runoff and less moisture in the soil in neighboring forests of the mountain. I mentioned clear-cutting practice. It's a very popular deforestation practice which devastates many acres of land, but is very beneficial for economic purposes. Low snowpack and high temperatures lead to drier soil and a record number of acres burned in wildfire. For example, the Owens Fire south of Hood River County, Oregon, which happened in 2014. Almost 600 wildfires across the nation have been caused by human interaction, so that is regardless of any wildfires that happen due to natural occurrences. So the problems presented by what I just mentioned are the decrease in snowpack leads to exceptional decrease in runoff during the summer season, as is so a reservoir of water for the drier seasons. Less moisture in the soil leads to an increase in fire, and therefore a decrease in usable soil organic matter. The three types of soil I mentioned again were old growth soil, clear cut soil, and burn soil. Here is the old growth soil that I collected, which has large evidence of moisture and fertility, while the clear cut and the burn show less water retention and overall just less fertility. In order to get a more accurate lay of the land, I suppose I just had to get outdoors and take a trip around the mountain, collecting various samples across, both near and far, in 3 different types of soils that I had to, that I was examining. And they were combined and filtered into a sample aggregate, as soil is a heterogeneous, non-uniform structure, so it's important to get a wide range and mixture of the substance. I took these samples per location and combined them into the aggregate and filtered 1 gram samples for 5 titrations per type of soil. The method of soil analysis I used was called the Walkley-Black titration method, which was a redox titration using the oxidizing agent potassium dichromate, which presents a very explicit and accessible calculation of % organic carbon, which you can see later. The reason why I chose this method was not only could I do it in my high school laboratory, but I couldn't use another method of carbon analysis, which is loss of ignition, which is basically burning soil samples and calculating the percentage of carbon from the mass remaining. But due to the presence of clay, which is a different kind of matter as compared to soil organic matter, it would present a different amount of percentage carbon. Also, this titration uses a very distinct color in the oxidizing agent, which shows difficulties in determining endpoint. These are the reactions that occur during the titration. So the first is the combination of chemicals and the soil in the oxidation of carbon with the reduction of the potassium dichromate. When the carbon is oxidized and therefore when the chromium is reduced, it goes from a color change of a bright orange to a dark blue-green. After that reaction is complete, we do a back titration, which is used to find the excess amount of chromium used to oxidize the carbon, and from that value we put it into this calculation, which again presents the organic carbon in percentage. So in simply put terms, carbon is oxidized by the potassium dichromate. Afterwards, we find the excess amount of chromate used in the oxidation, which through calculation, we find the total organic carbon oxidized. I did mention that the color of the potassium dichromate is a very distinct orange, and this is evident here with, in a blank titration value without any soil, but we do see when we put the soil, there is a dark orange and blue-green tint to it. On the left, you see my averages of the 5 titrations for each type of soil. In the old growth, we have a calculated average of around 60%, or 59.6 percentage carbon, and this is closer to the theoretical value of carbon found in soil, which is around 60%. But we do look at the 2 affected types of soil, which has a range of 15 to 30% organic carbon, which is around a 30 to 40% loss in comparison to the old growth. This is another example. This is the burn sample solution, and it shows a more distinct blue-green tint, which is closer to endpoint. Overall, the data does prove a significant loss of carbon from affected soils, while maintaining the fact that affected soils lost a significant amount of carbon in comparison to the old growth. Yet, in the 2 affected soils, we notice a margin of error overlap. A t-test shows significant difference between the 2 affected, between the old growth and the clear cut, the old growth and the burn, but an insignificant difference between the 2 affected. I would like to point out that there is still a value of organic carbon in the 2 affected soils, which proves that when human interaction affects a forest, the soil is not completely rendered useless. There is still a marginal amount of carbon remaining in the soil. So while there is no significant difference between the 2 affected soils, there is a significant difference between the 2 affected soils and the old growth soil. We do have to keep in mind that not all soil organic matter is lost, which is expected due to some debris remaining in my samples, and the time of impact of the burn, it did happen around 2 years ago, so there's a time for recovery of burned soil. As far as limitations and improving this experiment, taking more samples, having more titrations, and a wider expanse of data would definitely validate the samples, or it would improve the samples and the data collected. Taking in consideration the time of impact of the clear cutting and the wildfire, because of the recovery of soil, would be important to analyze, as well as the use of spectrophotometry to determine the endpoint. As I said, this method uses a very distinct color change, which is not easy to determine just with what's available to you in a high school lab. This experiment reinforces the understanding of what affects our soil in the Pacific Northwest. Human interaction, whether it is contribution to climate change, deforestation, or forest fires, will start long-term changes that will affect our local forests. This also raises an awareness of the beauty of our area, as the trees are not only just a resource for the economy, but they're what make the Pacific Northwest a truly beautiful place to live in. So the preservation of our forests and an awareness of what is happening in our climate is what will preserve the natural beauty of our area. I would like to acknowledge my chemistry teacher and mentor for this experiment, Ms. Gwendolyn Davis, who has supported me through this entire experiment. Access to chemicals and lab materials in Columbia River High School, as well as all of the other IB diploma students who have helped me with my experimentation. Here are my references, photo credits. And as we overlook beautiful Mount Hood, are there any questions?
Do you have an opinion on clear cutting versus other methods of harvesting trees?
The question was whether I have an opinion on clear cutting versus other deforestation methods. I believe that the lack of trees in forests will have the same effect. So I suppose as far as not just completely devastating entire lands and rather taking a smaller size of trees would make a difference, and in my opinion be better for the environment. But it's the fact that the deforestation is happening in the first place, which I would have a problem with. Thank you.
I may have missed it, so I'm sorry if I did, but how are you actually quantifying the colorimetric change in your carbon quantification?
The question was how I quantify the colorimetric change when doing my titration, correct? Well, I'll go back, hold on. So part of my calculation was a blank titration. So when the ferrous ammonium sulfate, which is the chemical I used to add during the titration, during the back titration, the color change was significant in this titration. It went to what my basis, I suppose, was for what the color change should be. So I used that chemical as a comparison for my other titrations. Are there any other questions?
I was a little unclear about when you gathered the soil samples, locations and aggregate things. Can you explain that a little bit more?
Certainly. So the question was my method of soil collection from these different locations and creating an aggregate. So I took all of the numbers there represent the locations to where I collected my soil. So I would also like to point out that I only collected soil from one burn site because of the limitations of accessibility. But from each location, I took careful measures to going near the trees and collecting 1 quart samples from each location and in different parts of each location. So when I say one location, I mean just location 1 or just location 2. And from location 1 and location 2, I took all 3 quart samples and combined it into a bucket and mixed it together to sort of homogenize it. And from that, I filtered the soil into 1 gram samples for my titrations. Does that answer your question? Thank you. Are there any other questions? The question was to elaborate on my first trial, which I suppose is an outlier to my data. I did mention that the theoretical amount of carbon found in soil is 60%, and this particular titration had a calculated around 90% organic carbon. Like I said, soil is not a homogeneous substance. It is not uniform, and it surely is not perfect. So the soil that I collected could have had a varied amount of carbon just in that single gram. So I did not omit this trial because I thought that I would see large maximums and minimums in all three, and it is important to include that in order, although it would sort of change my average, because at least the carbon is present in the soil itself. Thank you.
Just one question around disease. Tree disease, I think there's a lot of concern in whether it be old growth or new growth, but there's a lot of diseases that are killing off portions of the forest. Do you think it would be interesting looking at the component of the percentage of disease in some of these old growths? Do you think that will have an effect, and where would it stand compared to clear cutting or burn? I don't know, just wondering if you thought about disease.
The question was whether I had taken into consideration disease in old growth trees. I actually have not taken that into consideration, and I think it would be interesting to compare diseased trees as compared to, in regards to organic carbon in comparison to clear cut and burn. But if I had to make a guess, I still believe that any type of old growth forestry would have a higher amount of organic carbon in compared to forests that have been affected by human interaction. Thank you.